Welcome to the Educational Manual on Human Evolution, written on the basis of scientific conclusions presented in the research work Genetics of Human Evolution. Before you is a rigorous logical description of the monolithic construction of the physiological differences between humans and animals, confirmed by dozens of scientific works by geneticists and biologists. On the pages of this manual we will trace the key stages in the evolution of our species. Homo sapiens, the mutation of the uterine tube and uterus gene, the subsequent genetic reorganization by new genetic structures of the female central nervous system, and the restructuring of the entire body physiology of the offspring by fibroblasts of the central nervous system. Human differences from animals, expressed in the S-shaped form of the spine, the presence of the atlas and axis vertebrae, the displacement of the foramen magnum, the reconstruction of the jaw and vocal cords, as well as the rotation of the lobes of auditory and olfactory receptors toward the mammary gland zone, are consequences of mutational changes in the central nervous system. The evolutionary significance of these changes is as follows. The place of feeding of the newborn is genetically determined by the creation of one pair of mammary glands positioned as close as possible to the receptors of the head. Improved access of vision, hearing, and olfaction to the feeding area, to the mammary glands. Ensured physical orientation of the newborn's mouth toward the nipple of the mammary gland with the aid of vision and tactile receptors of the fingers. Enabled prolonged holding of the newborn in the mother's arms. Provided the newborn with constant protection from the external environment, pressing against the mother's skin ensured thermoregulation of the child's body. The appearance of pupillary lines on the naked skin of the fingers was accompanied by growth of the cerebral cortex. The new structural complexity of the brain became the foundation of higher nervous activity and the capacity for speech. Our research, supported by experiment and the necessary data from medicine, genetics, genome databases, paleontology, and archaeology, determined that human evolution represents the successful creation of a new biological construction in the form of an external uterine tube, EUT, copying the anatomy and function of the uterine tube during ovulation. The external uterine tube, EUT, is considered in this work as an evolutionary organ, mutation formed from the skin, muscular and neural structures of the skeleton and locomotor organs of the human ancestor according to the genetic template of the female reproductive system. Functionally, the structure designated by us as the external uterine tube may be interpreted as an analog of the mucous membrane of the uterine tube, formed by the skin of the hands and fingers. Using the method of comparative anatomy, Figures 1 and 2 demonstrate the presence in humans of an external uterine tube, anatomically represented by the system of upper limbs with pupillary skin matrices of the fingers, the cerebral cortex, hairless skin, and a flattened thoracic cage. The external uterine tube is homologous to the internal uterine tube, functionally reproduces its role in relation to the newborn, and is regarded as a mutational continuation of the female reproductive system extended beyond the boundaries of the human body. Let us examine the homology of the uterine tube and the external uterine tube in Figures 1 and 2. In Figure 2, the capture of the follicular oocyte by the fimbriae, its movement into the uterine tube and further toward the nutritive mucous membrane of the uterus, 1 asterisk, 2 asterisk, 3 asterisk, is shown. In Figure 1, the process of capturing the newborn by the mother's hands and transporting it to the source of nutrition, the mammary gland, 1, 2, 3, located on the pectoralis major muscle, is shown. Designations 6, 7, 8, 9 represent a complex of evolutionarily modified osteomuscular biological constructions, S-shaped spine, flexible articulation of the atlas with the axis, and the skull with displaced foramen magnum, evolutionarily modified to rotate the sensitivity lobes of the head receptors and the articulatory apparatus toward the infant feeding zone. 10, 5. The red dashed line indicates the area of child manipulation accessible to the anatomy and flexibility of the arms. The brown arrow in Figures 1 and 2 indicates the direction of transport of the oocyte from the ovary to the uterus and the transport of the newborn child from the place of birth. Common to both the uterine tube and the EUT is the presence of a biological directional vector for the movement of the oocyte and the newborn child from the place of origin toward the nutritive environment, as well as the presence of physical motor devices for the capture and transport of the oocyte and the newborn child. Let us examine the anatomy of the external uterine tube. In Figure 3, The external uterine tube, EUT, functions in relation to the newborn under any configuration of the mother's body, in a sitting, lying, or standing position. Its anatomical organization is represented by the system of upper limbs equipped with fine motor muscles that ensure the transfer of the newborn biological object from the birth canal to the mammary gland. 14. The hairless skin of the mother forms effective thermal contact with the newborn's body and provides skin lubrication through the secretion of sebaceous glands. The construction of the shoulder girdle and the joints of the upper limbs allows the arms to form a closed ring around the mammary glands located on the pectoralis major muscle, creating a zone of physical stability and protection for the newborn regardless of the mother's body position. The joint geometry of the arms, oriented both toward the birth organs and toward the mammary gland, ensures high adaptability of the external uterine tube to manipulations with the newborn. Figure 3 shows the anatomical correspondence of the forearm and elbow joint dimensions necessary to ensure contact of the pupillary matrices of the fingers with the nipple of the mammary gland. The position of the hands on the newborn's body and the spatial parameters of its body are tactilely determined by pupillary receptors, whereas conscious control of feeding and communicative interaction are realized by the central nervous system, the cerebral cortex, with the participation of visual, olfactory, auditory, and articulatory analyzers. During evolutionary transformations of the central nervous system, a new flattened osteomuscular organ was formed, the human face. This was achieved through reduction of the jaw apparatus and spatial reorientation of the olfactory analyzer on the frontal plane of the skull. 65. The facial muscles perform the function of expressing the mother's emotional states and participate in transmitting facial and gestural information to the newborn during feeding. Coordinated work of the facial muscles, the orbicularis oris muscle, gestures and facial expressions, combined with the acoustic parameters of speech, promotes focusing of the newborn's attention, development of its receptor systems, memory, reflexes, and the central nervous system as a whole, stimulating interest in feeding. Locomotion of the human quadrupedal ancestor was predominantly hunting-oriented and controlled by signals from the external environment. Cross movements of the limbs during walking and running were calculated mainly by the spinal cord. Evolutionary mutational processes led to the formation of a specialized, feeding locomotion for the arms with hands and fingers. In this system, the contours of the oocyte in the child's body serve as the supporting surface for the movements of the hands and fingers, while control and regulation of feeding are carried out through integration of visual, auditory, olfactory, muscular, and pupillary tactile signals. Figure 4 shows evolutionary anatomical features of the organs of smell, hearing, and vision that are present only in humans. The physiological rotation of these receptors toward the mammary gland created favorable conditions for the nursing mother to receive acoustic, olfactory, and visual signals from the external uterine tube. Designations: 1. New direction of the olfactory lobe oriented toward the mammary gland. 2. New direction of the auditory lobe oriented toward the mammary gland. 14. Formation of the human frontal part of the skull, face. 13. Adjustment of the ciliary muscle to incline the optical axis of the eye lens toward the mammary gland. 3. Foramen magnum displaced to provide better access of the head receptors to the mammary gland. 4. S-shaped spine increasing forward inclination of the skull with receptors in the sagittal plane. 5. Shoulder joint abducted from the mammary gland in the frontal plane by the human clavicle and scapula. 12. 6. Flexible elbow joint mechanism in conjunction with the hand. 7. Allows regulation of newborn capture and manipulation in three-dimensional space. 7. Hand with fingers and papillae. Greek small letter beta, angle of flexion, 0 degrees 170 degrees, and rotation, 0 degrees 180 degrees, of the elbow joint enables the mother to deliver the infant and transfer the newborn to the mammary gland for feeding. 8. Mammary glands created in maximum proximity to the head receptors. The nipple represents the specific feeding point, and the anatomy of the external uterine tube was structured in accordance with its location. Unlike animals with large jaws, a barrel-shaped thoracic cage, and forelimbs that block access of head receptors to the nipples in the human field of vision. With the presence of the atlas and axis and the displaced foramen magnum, lies the entire space in which feeding occurs. Through reconstruction of the forelimbs of the human ancestor into hands with papillae on the skin of the fingers, modernization of the elbow joint, and the action of the mutant central nervous system through its fibroblasts and the fibroblasts of the corresponding muscles of the spine, neck, and skull, the anatomy of the vertebrae was altered and maximally favorable conditions for control of the feeding process were created. We confirmed our theory of human evolution on 17 genes. We created a map of human evolution. We demonstrated that the evolution of butterflies, bees, ants, and termites followed a pattern similar to human evolution through a mutant ovipositor gene. Conclusion: applied significance of genetic research performed according to the theory of human evolution by duplicates of the uterine tube gene. Research options in the form of a specific long-term plan for conducting artificial evolutions using duplicates of the uterine tube gene and ovipositor gene in different species. Conducting secondary and tertiary artificial evolution in humans using duplicates of the gene of their own uterine tube. Expected result: creation of a new race, formation of new concepts regarding the potential cognitive capabilities of humans, reconsideration of the boundaries of intelligence, consciousness, and learning, as well as creation of a theoretical model of future humanity capable of mastering ultra-complex forms of thinking and responsibility. Conducting artificial evolution in mammals using duplicates of the gene of their own uterine tube. Expected result: bringing mammals to the cognitive level of humans, obtaining comparative data on which anatomical, neuronal, and behavioral changes are necessary for the emergence of complex forms of intelligence, thereby deepening understanding of the origin of humans as a biological species. Conducting artificial evolution in mammals using duplicates of the human uterine tube gene. Expected result: clarification of which elements of human evolution are unique and which can be reproduced in other biological systems, which has direct significance for the philosophy of anthropocentrism. Conducting artificial evolution in reptiles using duplicates of the gene of their own uterine tube. Expected result: expansion of the evolutionary worldview beyond the mammalian scenario and formation of a more universal theory of the emergence of intelligence. Conducting artificial evolution in reptiles using duplicates of the butterfly ovipositor gene. Expected result: induction of a complete metamorphosis in reptiles similar to that of a butterfly, acquisition of new knowledge about the possibility of radical phase transitions in organismal development, which is significant for understanding saltational evolutionary changes. Conducting artificial evolution in insects using duplicates of the human uterine tube gene. Expected result: obtaining insect species with increased intelligence and unusual physiology, identification of fundamental limitations and universal principles of evolution applicable to all forms of life regardless of their size and organization. Conducting artificial evolution in cetaceans using duplicates of the human uterine tube gene. Expected result: creation of a large mass aquatic human and the dolphin-like human, development of a theoretical model of aquatic anthropomorphic intelligence with significance for astrobiology and the search for intelligent life in the universe. Conducting combinations of artificial evolutions in cartilaginous fishes, sharks, rays, and bony fishes, sturgeons, salmon, perches. Expected result: creation of a new lineage of cartilaginous species, restoration of the evolutionary continuity of Earth's biosphere, and deepening understanding of the origin of complex ecosystems. Conducting combinations of duplicates of uterine tube and ovipositor genes in lower primates, rare and exotic animals, as well as species with pronounced immune resistance. Expected result: obtaining a new species immune fluids in blood suitable for use as medicinal substances, identification of fundamentally new molecular and physiological solutions capable of influencing the development of medicine, immunology, and the biological security of humanity. Final perspective. Feeding and studying the obtained forms in a controlled scientific environment makes it conceptually possible to consider the creation of an evolutionary scientific zoo, a Babylon of possible life forms, serving not demonstration but knowledge. Such an approach expands understanding of what forms of intelligence and life may exist in the universe and defines a new place for humans as investigators of evolution, not merely its result. Conclusion: in the work Genetics of Human Evolution, the possibility of genetic duplication and mutation of the fallopian tube gene and the influence of this gene on the central nervous system of the human ancestor during meiosis were studied. On the basis of this conclusion, genes were identified whose tissue and cell expression peaks, displayed in histograms of the musculoskeletal system and reproductive organs of the female mammal and unified by the modular method, provide a detailed picture of human evolution.